Hello, everyone. Welcome to today's live broadcast, Thiol-linked alkylation for the metabolic sequencing of RNA. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Lexogen. Lexogen is a transcriptomics and next-generation sequencing company focusing on the development of technologies for complete transcriptome sequencing. Its portfolio includes innovative molecular biology kits, software, and services for RNA-seq. To learn more, visit www.lexogen.com. So we have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Stefan L. Ameres, PhD, Group Leader of the Institute of Molecular Biotechnology in Vienna, Austria. Dr. Ameres acquired his master's degree in biology at the Friedrich-Alexander-University, Erlangen-Nuremberg. He did his PhD thesis in the lab of Renée Schröder at the Max F. Perutz Laboratories of the University of Vienna. There, Dr. Ameres performed the first in-depth enzymatic characterization of the human RNA interference effector complex. Next was a postdoc with Phillip Zamore at the University of Massachusetts Medical School, where he focused on the small RNA-mediated gene regulation in flies and mammals. Dr. Ameres started his own lab in 2012 at the Institute of Molecular Biotechnology in Vienna. His lab investigates the mechanism and biology of post-transcriptional gene regulation, supported by the European Research Council through an ERC Starting Grant. Dr. Ameres is an elected Young Academy member of the Austrian Academy of Sciences and recently selected EMBO Young Investigator. His complete bio is found on the LabRoots website. I will now turn it over to Dr. Ameres for his presentation.
Thank you very much, Judy. So today I would like to introduce you to a novel gene expression profiling protocol that we have developed at the Institute of Molecular Biotechnology at the Vienna Biocenter in Austria, which adds a novel dimension to the analysis of standard high-throughput RNA sequencing approaches. And this is the dimension of time, which enables to gain insights into how gene expression programs are actually established in order to study the regulation of gene expression at the transcriptional as well as at the post-transcriptional level. So for cells to fulfill their biological role as they give rise to the different cell types in the context of tissues and organs of a multicellular organism, they need to mount specific gene expression profiles, and failure to do so can result in severe diseases such as cancer. So gene expression profiling by high-throughput sequencing has really revolutionized the way we can associate distinct gene expression profiles to cellular states, thereby providing mechanistic entry points to understanding the basis of physiological as well as pathophysiological processes at the molecular level. However, currently available technologies for the high-throughput profiling of gene expression really only take a snapshot of the relative abundance of any given transcript at a given time point inside cells. What they don't do is to provide us with information on how this gene expression profile is actually established, which means the rate at which each individual transcript is produced and the rate at which it is degraded. But it is actually this information that provides us with insights into how each transcript is regulated, both at the transcriptional level, which is determined by a combinatorial action of promoters, enhancers, associated transcription factors, or chromatin landscape, to just name a few, as well as at the post-transcriptional level, which defines transcript stabilities as determined by RNA binding proteins, RNA modifications, small RNAs, RNA structure, codon usage, and many more. Now, because of the importance of understanding the molecular basis for gene regulation, several approaches have been developed to globally address the kinetics of gene expression at the genomic scale. For example, genome-wide analysis of nascent RNAs engaged with transcriptionally active RNA polymerase by high-throughput sequencing, such as through approaches like GrowSeq, ProSeq, or NetSeq, represent powerful methods to investigate transcriptional activity. And despite their utility, these approaches are really technically challenging and not readily scalable, and they do not discriminate functional RNA molecules that are fully processed from transcriptional byproducts. On the other hand, RNA decay rates are commonly determined by globally interfering with RNA polymerase, followed by the relative quantification of RNA molecules over time. But due to the global loss of RNAs, the quantification of absolute RNA half-life remains imprecise. Furthermore, inhibiting polymerase II activity compromises cell viability and induces major stress responses, triggering secondary effects, including the selective stabilization of specific transcripts. Now, in principle, metabolic RNA labeling approaches that employ the incorporation of nucleotide analogs enable to track RNA species over time without interfering with cellular integrity. Among these, 4-thiouridine is probably the most widely used nucleotide analog to study the dynamics of RNA expression, because it is readily imported into many metazoan cells by equilibrate nucleoside transporters and provides unique physicochemical properties for thiol-specific reactivity and affinity, which enables, for example, the biochemical separation by reversible biotinylation. And indeed, such approaches have been successfully applied to cultured cells of diverse biological origin and across many species through methods such as S4U-seq or more recently TT-seq. However, like any biochemical separation methods, the underlying protocols are laborious, require ample starting material, and typically encounter the problem of low signal-to-noise performance, in part because of limited biotinylation efficiencies. And again, such strategies are really not readily scalable. Furthermore, the analysis of labeled RNA transcripts by biochemical fractionation, followed by sequencing, requires extensive controls. So one really needs to sequence 3 samples per time point, the input, the fractionated RNA, as well as the flow-through, in order to provide integrative insights into gene expression dynamics. And it fails to report global effects unless spiking strategies are applied. Now, to overcome these limitations for measuring the intracellular kinetics of RNA transcription and turnover, we have developed a novel protocol which we refer to as Thiol-linked alkylation for the metabolic sequencing of RNA, or in short, SLAM-seq. Now, how does SLAM-seq work? So we employ the well-established metabolic labeling strategy employing the nucleotide analog 4-thiouridine, which, as I've mentioned, is readily taken up by cultured cells and incorporated into newly transcribed RNA. So total RNA is then prepared at given time points after labeling, and instead of biochemical fractionation, SLAM-seq employs a thiol-specific chemical modification strategy. To this end, we use iodoacetamide, which specifically reacts with a primary thiol group present in 4-thiouridine, but not any other naturally occurring nucleotide. This alkylation reaction modulates the base pairing capacity of 4-thiouridine, so that when RNA is afterwards subjected to cDNA library preparation for high-throughput sequencing, reverse transcriptase will misincorporate in this process a guanine across chemically modified 4-thiouridine, which upon high-throughput sequencing enables to biothematically detect 4-thiouridine labeled transcripts by T to C conversions. Now, what I would like to do in this webinar is to show you how SLAM-seq enables cost-effective insights into intracellular RNA kinetics at really an unprecedented accessibility, efficiency, and scalability, and therefore represents a powerful tool for the dissection of fundamental biological mechanisms that control gene expression. So before I do so, I would like to highlight the people in the lab who really established SLAM-seq. And this project was led by an outstanding postdoc in the lab, Veronika Herzog, supported by a PhD student, Brian Reichhold. Busche Butt is a PhD student who was involved in some of the bioinformatics work, And although I will not go into detail in this presentation about the bioinformatics background in analyzing the data, I would like to highlight two very gifted bioinformaticians who developed a sophisticated bioinformatics pipeline that enables the rapid analysis of SLAM-seq datasets, even for bench scientists. That is Tobias Neumann from the Institute of Molecular Biotechnology and at the Zuber Lab, as well as Phillip Zamore from Arndt van Hesseler's lab at the Perutz Laboratories, both located at the Vienna Biocenter. Now, I would like to use the next couple of slides in order to give you, to guide you through the key steps of the SLAM-seq protocol and the experiments we have performed to validate the chemistry and the nucleotide conversion readout. I will then introduce you briefly to the library preparation methods we are employing and the important technical considerations that need to be taken into account when starting a SLAM-seq experiment. And in the end, I would like to show you briefly how we employ SLAM-seq in mouse embryonic stem cells to get insights into the regulation of pluripotency-associated gene expression at the transcriptional and post-transcriptional level. Now, first, we have to validate the key steps of the SLAM-seq protocol in order to ensure that this is a highly quantitative method. First, in order to provide rapid and direct experimental access to metabolically labeled RNA by sequencing, we employed a chemical derivatization approach for 4-thiouridine. To this end, we employed the primary thiol-reactive compound iodoacetamide, which covalently attaches a carboxyamido methyl group to 4-thiouridine by nucleophilic substitution. We confirmed the quantitative alkylation of 4-thiouridine by two approaches. First, by spectrophotometry, where we monitor the shift in the characteristic absorbance spectrum of 4-thiou from 335 nanometers, as shown in the left, to 297 nanometers upon iodoacetamide-directed alkylation, and found that under optimal reaction conditions, absorbance at 335 nanometer decreased 50-fold compared to untreated 4-thiou, resulting in close to complete alkylation within 15 minutes. And we validated these results by mass spectrometry, which confirmed close to complete derivatization efficiencies. Now, in a second important point, the quantitative identification of 4-thiou by sequencing presumes that reverse transcriptase passes alkylated 4-thiou residues without drop-off. We therefore determined the effect of 4-thiou alkylation on RT processivity in primer extension assays using radioactively labeled primer and synthetic RNA that contains a single 4-thiouridine at a defined position. So we determined this before and after treatment with iodoacetamide, and then assessed the products of the reverse transcription and possible drop-off signal by high-resolution gel electrophoresis. So when we zoom into to the region where the 4-thiou is present, compared to the control, then compared to an identical RNA without 4-thiou incorporation and normalized to background drop-off signal, we did not observe a significant effect of 4-thiou alkylation on RT processivity when using any of three different commercially available reverse transcriptases, as shown by the quantification in the left. So to evaluate the effect of 4-thiou alkylation on RT-directed nucleotide incorporation, we isolated the full-length products of primary extension reactions, PCR amplified the cDNA, and subjected the libraries to high-throughput sequencing. Again, we used the non-4-thiouridine labeled synthetic RNA as a control and found that treatment of RNA with iodoacetamide has no adverse effect on mutation rates of any given nucleotide in the control oligo, as shown in the quantifications to the left. While the presence of 4-thiou in a control RNA prompted a constant 10 to 11% of T2C conversions, as seen at position 9, this is presumably due to base pairing variations of 4-thiou tautomers. 4-thiou alkylation increased T2C conversions by 8.5-fold, resulting in a conversion rate of greater than 94%. So together with the chemical modification efficiency that I've shown before, we concluded that uroacetamide treatment followed by reverse transcription specifically identifies 4-thiol incorporations in RNA at single nucleotide resolution by sequencing at an efficiency of greater than 90%. Now, to combine SLAM-seq with a rapid, robust, and cost-effective high-throughput RNA sequencing strategy, we employed the mRNA 3' sequencing method called QuantSeq. Now, just very briefly, the way 3' mRNA sequencing works is that total RNA is subjected to reverse transcription using an anchored oligo-DT primer, which primes the reverse transcription at the beginning of a poly-A tail of RNA pol2 transcripts. After the reverse transcription, RNA is removed, and second-strand synthesis is directed by a randomized oligo containing an adapter. And after purification and PCR amplification, these libraries can be subjected to high-throughput sequencing using the Illumina platform. Now, this sequencing approach has several key advantages, which I will briefly go through. First of all, it uses total RNA as input, therefore does not require prior poly-A enrichment or rRNA depletion. The library preparation follows a robust and rapid workflow, which enables to generate sequencing-ready libraries in less than 5 hours, and this can be readily scaled through automated library preparation protocols. It provides exceptional strand specificity, and it identifies transcript 3' ends, which enables to de novo annotate transcript ends, which is highly recommended, and we have developed an algorithm to do that from QuantSeq data that for which you find the reference indicated in the respective point. It also enables to do simple gene counting, so that means just one fragment per transcript is produced, therefore normalization to gene length is obsolete. And the lower library complexity further enables to do less deep sequencing and therefore enable cost-saving multiplexing. It also facilitates the bioinformatic analysis because junction detection is not required, because reads are generated from the transcripts at the 3' end where nearly no junctions are located. And finally, a SLAM-seq specific advantage is that at the high coverage of mRNA 3' tags facilitates the calling of TC conversions, particularly because amplicons derive from 3' UTRs, which are inherently AU-rich. There are, of course, also some limitations. It is, of course, restricted to polyadenylated RNA species, so therefore RNA pol2 transcripts, and it does not provide information on splice isoforms or transcript start sites. Now we have performed validations of QuantSeq across close to 100 libraries, meanwhile, mostly in mouse embryonic stem cells. And the meta-analysis, as you can see here, shows a clear enrichment of the fragments within around 250 nucleotide window upstream of annotated mRNA 3' ends. And comparison of QuantSeq signal shows an overall high and significant correlation with standard RNA sequencing approaches across more than 17,000 transcripts. And most importantly, QuantSeq delivers highly reproducible results across biological replicates. Now, having spent this detour on the library preparation protocol, I would like to now go into some sanity checks that we highly recommend to implement before starting a SLAM-seq experiment to be sure that one's working under unperturbed conditions and in an effective way. First of all, we highly recommend to titrate 4-thiol into the cell type of interest to determine toxic effects, which typically set in at very high 4-thiol concentration, presumably because in this scenario 4-thiol results in rRNA biogenesis defects that have been reported before. We therefore typically work at concentrations far below the EC50 and recommend as an ideal concentration the EC10 value as a reasonable upper limit. Now, because the uptake of 4-thiol can vary quite substantially between different cell types, we consider this to be an essential first step in setting up this method. And second, before going into library preparations, we recommend to test uptake of 4-thiouridine and incorporation into RNA, which can be rapidly assessed after total RNA preparation and single nucleoside digestion, followed by HPLC analysis as previously described as shown in the citation. For this, we recommend to label to saturation, that means for 12 to 24 hours, because the majority of transcripts in total RNA consists of very stable non-coding RNA species, including rRNA and tRNA. So to test if SLAM-seq enables us to quantify 4-thiol-labeled RNA pol-2 transcripts in mouse embryonic stem cells, we subjected the cells to 4-thiol labeling for 24 hours and prepared QuantSeq libraries after chemical modification using the SLAM-seq protocol. The whole library preparation is extraordinarily efficient and takes less than 5 hours, including the chemical modification step. Now, how do the results look like? Here you can see a genome browser screenshot of a particular gene called TRIM28, where we compare regular RNA-seq, so whole transcript RNA sequencing approach in the top, a CAP-seq track in the middle, which marks the transcript 5' end, and QuantSeq on the bottom, which shows a pileup of reads at the transcript 3' end as expected. When we zoom into this region and inspect the underlying, or a subset of the underlying reads, we observe a low mutation frequency in samples generated from untreated cells, and after metabolic labeling for 24 hours, followed by SLAM-seq, we observe a strong accumulation of TC conversion containing reads. So meta-analysis show that these TC mutations distribute evenly across the area covered by sequencing using QuantSeq, and when we compare all possible mutation frequencies, we find that mutations are found below the sequencing error reported for Illumina technology, which is below 0.1%, and only upon metabolic labeling with 4-thiol shown on the right, we observe a statistically significant increase only in TC conversions, but not any other nucleotide conversion. Note, please, the broken y-axis to interpret the full range of the signal, which spans 2 orders of magnitude. Now, together, these analyses confirm that SLAM-seq provides a robust access to 4-thiol incorporation events in mRNA following 4-thiol metabolic RNA labeling in cultured cells. Now, as mentioned in the introduction, steady-state expression, gene expression, is dictated by the relative contributions of the rate of transcription and RNA decay, 2 parameters that cannot be derived from standard RNA sequencing approaches. Now, to test if SLAM-seq provides quantitative access to the polyadenylated transcriptional output, we subjected mouse embryonic stem cells to a short 45-minute 4-thiol pulse labeling, followed by total RNA extraction, alkylation, and mRNA 3' end library preparation. We first approximated the number of molecules that were made within the short time frame of labeling by inspecting background error subtracted T2C conversion containing reads for individual transcripts as displayed here. Indeed, initial inspection of these selected transcripts with comparable steady-state abundance revealed that transcript-specific differences in the number of recovered TC reads. While high levels of T2C conversions were recovered for the embryonic stem cell-specific transcription factor SOX2 and the inherently unstable primary microRNA transcript derived from the miR290 to 95 cluster, the housekeeping transcript GAPDH associated with fewer TC reads, presumably because its accumulation does not require high transcriptional output, but instead relies on high transcript stability. Now, transcriptional output by RNA polymerase II is regulated by transcription factors that bind cis-acting regulatory elements known as enhancers to recruit co-activators and pol-2 to target genes. So in mouse embryonic stem cells, the pluripotency stage is largely governed by a small number of master transcription factors, including OCT4, SOX2, and NANOC, which drive the expression of target genes necessary, shown here in blue, necessary to maintain the embryonic stem cell state, and that positively regulate their own promoters, forming an interconnected autoregulatory loop. Now, transcriptional output measurements by SLAM-seq revealed that OCT4, SOX2, NANOC target genes, shown in blue and red, produced overall a larger amount of T2C conversion containing reads in the 4-thiol pulse experiment, confirming their high transcriptional activity. Now, to connect these observations to molecular principles of transcriptional gene regulation, we correlated the measured transcriptional output two proximal enhancer loci, which can be classified by their association with the transcription factors OCT4, SOX2, and NANOC. We call these OSN enhancers, as displayed here. This has previously been described by ChIP-seq approaches. Now, transcripts derived from approximately 2,000 expressed genes with proximal OCT4, SOX2, NANOC occupancy, shown as OSN, produce significantly higher levels of T2C conversion containing reads upon pulse labeling when compared to close to 5,000 genes without proximal OCT4, SOX2, NANOC enhancers, shown as no. A subset of enhancers in mouse mRNA stem cells were previously described to form arrays of concatenated regulatory elements, also known as super enhancers or strong enhancers, here labeled as SE, with unusually strong accumulation of transcriptional co-activators, specifically mediator. In fact, the 150 genes, around 150 genes, in the proximity of such strong enhancers exhibited the highest transcriptional outputs, significantly offset from typical enhancers in our SLAM-seq measurements. Now, together with the fact that we detected a significant correlation between transcriptional output measurements determined by SLAM-seq and alternative methods, like, for example, GrowSeq, these data confirm that short 4' pulse labeling in combination with SLAM-seq and mRNA 3' sequencing enables to uncouple the immediate transcriptional output from stability effects to directly measure the functional POL2-derived transcriptional activity at the genomic scale. The method therefore provides a rapid and scalable approach to study transcriptional gene regulatory circuits in mammalian cells. Now, in the last part, I would like to illustrate how SLAM-seq can be employed for the direct measurement of mRNA transcript stabilities. Now, to address this, we subjected mouse embryonic stem cells to 4' metabolic labeling for 24 hours, followed by a washout and chase using non-thiol containing uridine, and prepared total RNAs at various time points along the chase. Now, total RNA was again subjected to alkylation as described before and mRNA 3' sequencing. So first again, let's inspect candidate genes. Here you can see a genome browser coverage track for 4 different genes with a zoom in to their 3' ends across the whole time course. Now, when we inspect all reads, that means a TC conversion containing and non-containing reads, we observed that steady-state gene expression stayed constant for these individual transcripts across the whole time course, which is what one would expect if we were working under unperturbed conditions. And this is also confirmed globally when comparing steady-state expression globally along the time course. Now, in contrast, TC conversion containing reads, shown in red here, decreased over time in a transcript-specific manner, which is independent of the steady-state abundance of each transcript. After calculating the background subtracted U content and coverage normalized TC conversion rate for each transcript at every time point relative to the onset of the chase, we observed that conversion rates fit well to single exponential decay kinetics, enabling the robust determination of polyadenylated transcript half-lives. As expected, RNA stabilities differed by more than 1 order of magnitude among individual transcripts. A very short half-life was, for example, observed for the primary microRNA transcript derived from the mu290-95 cluster, which has a half-life of less than 1 hour. Most certainly because it is subjected to rapid co-transcriptional processing by the RNase3 enzyme DROSHA in the nucleus. Now, by fitting the data to 8,400 transcripts with a steady-state expression above a certain cutoff to single exponential decay kinetics, we determined a median mRNA half-life of 3.9 hours, corresponding to a cell cycle normalized half-life of 4.3 hours, which fall within the range of previously determined mRNA stabilities in mammalian cells, ranging in median half-life from between 3 and 10 hours, depending on cell type and experimental technique. Now, because we use mRNA 3' end sequencing, we can even assign transcript half-life to each of the 3' isoforms of any given gene locus. As illustrated here, for this gene RFRP1, where we can see a more than 5-fold difference in mRNA 3' isoform stability. This screenshot also illustrates the importance of re-evaluating gene annotations with respect to their annotated 3' ends, because many genes produce cell type specific variants which warrant de novo annotation. Here is another example for the gene TAX261, which produces 3 3' isoforms where the two longer variants are half as stable when compared to the shorter, and the longest isoform even extends beyond the REF-seq annotation, but shows clear connectivity to the upstream transcription unit when inspecting whole transcript-derived RNA-seq signal shown on the very top. Now, previous investigations of mRNA stability proposed a close relationship between transcript-specific mRNA half-life and the encoded physiological function. For example, when we rank the more than 6,600 transcripts for which half-life was determined at high accuracy, according to their relative stability, and performed gene ontology analysis for the 10% most or least stable mRNAs, transcripts with short half-lives significantly enriched for regulators of Pol2-dependent transcription, while stable mRNAs, shown in red, associated with the gold terms translation, respiratory electron transport, and oxidative phosphorylation. So this means that transcripts encoding proteins with housekeeping functions, such as protein synthesis or respiration, tend to decay at low rates, which perhaps reflects the evolutionary adaptation to energy constraints. So an mRNA that is needed all the time is rendered stable in order to not have to produce it over time, over and over again. In contrast, transcripts with a regulatory role, such as transcription factors or cell cycle genes, tend to decay faster, most certainly because control over the persistence of genetic information facilitates adaptation to environmental changes. So that means upon external stimuli, these regulatory genes must be turned off rapidly, and this is implemented at the RNA level by having intrinsic short half-life, low stabilities. Now, to further validate SLAM-seq, we performed mechanistic studies on a specific post-transcriptional gene regulatory pathway with well-established biological function in mouse embryonic stem cells. To this end, we focused on microRNAs, which act as key regulators of gene expression in a variety of biological contexts. In mouse embryonic stem cells, these microRNAs contribute to cell state maintenance and transitions by tuning the expression of ES cell transcripts and promoting their clearance during differentiation. At the molecular level, microRNAs are short, around 22 nucleotides, non-coding RNAs that act as guides for ribonucleoprotein complexes that target complementary sites, usually within the 3' UTR of mRNAs. Now, microRNAs elicit their function by repressing either translation or promoting mRNA decay, although the relative contribution of repressive modes remains a matter of debate and may vary in different biological contexts. Now, to estimate the effect of microRNAs on mRNA stability in mouse embryonic stem cells, we focused on the microRNA family shown here, the MIR291A family, which share the same C sequence and target repertoire and represents more than half of all small RNAs expressed in this cell type, as shown in the abundance plot derived from small RNA sequencing on the right corner. So we first investigated the general association of microRNA targets with mRNA stability. And we found that with a median half-life of 2.9 hours, mRNAs that were predicted to be targeted by this microRNA family, MIR291A family, that means they harbor sites complementary to the microRNA in the 3' UTR, were significantly less stable, shown in red, compared to transcripts that contain no target site, shown in black, which show an average half-life of 4 hours. Now, transcripts with conserved sites, so microRNA binding sites that are conserved throughout evolution, exhibited even shorter half-lives with a median half-life of 2.6 hours. Now, this is, of course, only associative, so to confirm direct contribution of microRNAs to transcript destabilization, we determined changes in mRNA half-life in mouse embryonic stem cells depleted of the core microRNA biogenesis factor exportin 5, which was depleted by CRISPR-Cas9 genome engineering. When compared to wild-type cells, depletion of expo5 significantly decreased overall microRNA levels by more than 90%, and particularly the miR-291A family members by more than 95%, as determined by spike in normalized small RNA sequencing. Now, consistent with a function of microRNAs in triggering mRNA decay, we observed a significant increase in relative mRNA stabilities for targets of miR-291A family when compared to transcripts containing no target sites upon depletion of exportin 5. Now, target binding by microRNAs is primarily mediated via the so-called seed sequence, which encompasses nucleotides 2 to 7 or 8 when counted from the microRNA 5' end. And it has previously been proposed that the efficiency of microRNA-mediated gene regulation can be classified according to the extent and nature of base pairing. To get further insights into this, we inspected the efficacy of 4 different site types, 6 MER sites, 7 MER with an A across position 1, 7 MER with a match at position 8, and 8 MER sites, and investigated the effect on mRNA destabilization. While each site type responded to exportin 5 depletion with a significant increase in mRNA stability, relative to untargeted transcripts, 6 MER sites clearly showed the weakest effect, followed by the 2 7 MER site types, and 8 MER sites showed strongest de-repression, confirming that classification of microRNA site types translates into the biological efficacy of microRNAs. And it highlights the accuracy at which SLAM-seq protocols enable to measure even minute differences in mRNA stability. Now, in summary, these data provide a quantitative evidence for microRNA-mediated mRNA decay in mouse embryonic stem cells and validate SLAM-seq as a sensitive tool to study the molecular basis underlying the fine-tuning of mRNA expression levels by microRNAs, and therefore also more generally the mechanisms underlying post-transcriptional gene regulation. Now, with this, I would like to come to the summary of today's presentation. So I've introduced you today to a novel high-throughput sequencing method for the global metabolic sequencing of RNA, which we refer to as Thiol-linked alkylation for the metabolic sequencing of RNA, or SLAM-seq. SLAM-seq is an orthogonal chemistry-based sequencing protocol that uncovers 4-thiouridine incorporation in RNA species at single nucleotide resolution and close to absolute stoichiometry in the context of unlabeled RNA. Now, when coupled to well-established 4-thiou metabolic labeling protocols in cultured cells and coupled to mRNA 3' sequencing, SLAM-seq enables rapid access to gene expression dynamics in living cells at unprecedented accessibility, efficiency, and scalability. While the method is in principle compatible with any RNA library preparation method that involves cDNA conversion, I've shown you that coupling it to mRNA 3' sequencing via the QuantSeq approach provides some key advantages, including that it samples polyadenylated RNA species in the context of total RNA. It provides further insights into 3' isoform-specific expression dynamics. It facilitates bioinformatic analysis due to molecular counting approaches, and it has the key advantage of producing highly reproducible gene expression profiling libraries through a rapid workflow. In principle, this approach can also be applied with very low input material, down to 100 picogram, although we have standard used 500 nanogram as input material. I think it's worth to mention because in many of the experimental setups, the input material is actually limiting. So we think that this approach is covering this aspect. So at the biological side, I've shown you some of the applications of SLAM-seq. On the one side, I've shown you that short 4-thiou pulse labeling followed by SLAM-seq enables to measure functional processed RNA pol II directed transcriptional output and quantifies transcriptional gene regulation as confirmed by mechanistic studies on enhancer function in mouse embryonic stem cells. And I've also shown you that fourth IU pulse labeling followed by washouts and chase experimental setup enables to directly measure genome-wide transcript stabilities in order to dissect the molecular basis for post-transcriptional gene regulation, as exemplified by the function of microRNAs, which mediate site-type dependent transcript destabilization in mouse embryonic stem cells. Now, the people involved in this work I've already highlighted in the beginning. Once again, this is mostly led by a great postdoc in the lab, Veronika Herzog, supported by two PhD students, Brian Reichholz and Pusha Butt. We had great collaborations here at the Vienna Biocenter with Johannes Stuber's lab and Anne van Heseler's lab, mainly the bioinformaticians who contributed to a rapid pipeline that facilitates the analysis of SLAM-seq, Tobias Neumann and Philipp Reschennieder. The work benefits enormously from outstanding service facilities at IMBA and the Vienna Biocenter, and I would like to particularly highlight the exceptional available career opportunities here on campus, especially through the International BBC PhD program. And work of this project was funded by the European Research Council and the Austrian Science Fund. So at the end, I would like to briefly mention that SLAM-seq is a licensed technology that is commercially available from Lexogen. They provide kits that facilitate the setup of SLAM-seq experiments through a SLAM-seq Explorer kit that includes a cell viability titration module for assessing 4th-IU toxicity in target cell lines and a 4th-IU incorporation module to measure 4th-IU uptake and incorporation rates using HPLC. And again, we highly recommend to perform these control experiments because of reasons that I mentioned in the talk. And they also provide SLAM-seq kinetic kits that enable anabolic, on the one side, anabolic labeling experiments, which label newly transcribed RNA with 4th-IU and measure nascent RNA expression to analyze RNA synthesis kinetics, and catabolic kits on the other side that labels existing RNAs in order to assess transcript stability for the analysis of RNA degradation kinetics. And finally, for the rapid bioinformatics analysis, the SLAM-seq datasets of SLAM-seq datasets we have developed in collaboration, as I mentioned, with Tobias Neumann and Philipp Reschennieder, a bioinformatic pipeline called Digital Unmasking of Nucleotide Conversion Containing K-MERs, or DUNC, which as SLAM-DUNC has been included on Bluebee cloud-based platform and is accessible with a special code offered by Lexogen. So with this, I would like to thank you very much for your attention, and I would be happy to answer any questions.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Dr. Stefan Ameres is now joined by Lexogen application scientist Dr. Stephanie Bannister to answer your questions about the SLAM-seq technology and the kits. And our first question is, does SLAM-seq work well across different cell culture types, for example, primary neuronal culture? And that would be for Stefan.
Yes. So we have tried multiple different cell lines meanwhile, and we find that in average across different mammalian cell types that we've tested, we find that all of them do take up 4th-IU uridine, but they do so to varying degree. So I think it is very wise to start a SLAM-seq experiment by just testing 4th-IU uptake by first measuring toxicity and using, for example, the single nucleoside digestion in HPLC approach from total RNA, which doesn't require basically a lot of time and effort to investigate whether 4th-IU is taken up and incorporated into newly transcribed RNA in the cell type that is studied. So this is a setup that can be found also in our publication, I think, and is available through the Lexogen, that I would definitely recommend before starting a SLAM-seq experiment in a novel cell type that has not previously been carefully assessed for its ability to take up and incorporate thiouridine. But overall, I'm very positive, so we haven't seen dramatic outliers so far, so cell types that are not at all taking up thiouridine.
Stefan, this next one is for you too. For human cells, for example, HEC293 or HeLa, to what depth would you need to sequence if using a short 4SU pulse around 10 minutes, given that there is no enrichment for labeled RNA, which would be a small fraction of the total RNA?
So that is a very good question. So this is so I mentioned that how SLAM-seq uncovers newly transcribed RNA is that it does this in a context of unlabeled RNA species, which is an advantage on the one side and a disadvantage on the other side, because in short pulse labeling approaches, of course, a very small fraction will be labeled. But we've done now various experiments using short pulse labeling, and in average, a good approximation for the sequencing depth is that one can sequence up to 3 multiplex samples in one lane of single read 100 on an Illumina HiSeq 2500 platform, for example. That will enable to analyze transcriptional output of thousands of genes. So that's an approximation that I can give. But of course, again, also in this experimental setup, I would recommend to optimize for thiou labeling conditions by using maximum tolerated amount of thiou, which will of course also facilitate then, in particularly in short pulse labeling approaches, the ability to uncover de novo transcribed transcripts.
Stefan, what is the future of metabolic RNA labeling in vivo? Can SLAM-seq or similar methods be tweaked for in vivo?
That is a very good question. So there are, of course, approaches going on to try to implement SLAM-seq in similar approaches in vivo. Metabolic labeling in vivo can be achieved. So studies have shown that thiou can be injected in animals, and even using a specific enzyme that converts, for example, uracil and uridine, called UPRT, one can perform tissue-specific labeling of RNA. So labeling is possible in vivo. Whether kinetic gene expression analysis, as opposed to simple tissue-specific gene expression analysis, is really possible depends on pharmacokinetics of thiou. So how rapidly does actually a given cell type take up thiouridine, and to time these experiments to really get to gene expression dynamics in vivo will be challenging in future.
This next one is for you too. I would like to know the cell line that you have used and the percentage of 4SU incorporation in your total RNA. Thanks.
So the percentage of thiou, if I understood that correctly, the percentage of thiou incorporation will again depend on the labeling efficiency. What we have determined is we have compared after saturation labeling basically how many transcripts that incorporate the thiou we can actually uncover. And for this we have basically compared a poly-A pull-down and mass spectrometry, which gives one estimate of how many thious are replaced by uridine, and SLAM-seq on the other side, which give basically both the same results, which is that in mouse embryonic stem cells, every 40th nucleotide, every 40th uridine, is replaced by a thiouridine. And when one translates that to mRNA 3' sequencing using the QuantSeq approach, for example, then and one uses single read 100, then this would enable to recover more than 70% of all labeled transcripts. So a significant fraction of labeled transcripts is directly measured through this approach by high throughput sequencing. I hope this answers the question.
Stephanie, this next one is for you. What kits do I need to purchase for my first experiment with SLAM-seq?
Thanks, Judy. So as Stefan just mentioned, the ideal way to start with a SLAM-seq experiment, especially when using a new cell type or with a different labeling approach, labeling duration for different experimental setups, is to start with the SLAM-seq Explorer kits. So we have two modules from the SLAM-seq Explorer kit. The first one is the cell viability titration module, and this allows you to assess the S4U, 4-thiouridine toxicity in the target cell line that you're working with. And this provides all of the labeling S4U reagent and allows you to optimize those labeling concentrations for your kinetics experiments. And then once you have established the cell titration toxicity levels, you can go ahead with the S4U incorporation module, which is the other Explorer kit, and this allows you to measure the S4U uptake and the incorporation rates using the HPLC analysis method that Stefan has described. And once you have these two parameters in place, so you know the optimal concentration for your labeling experiments, and this allows you to understand the incorporation rate, and so that the labeling percentage in your samples, then you can proceed with kinetics experiments. So we have two kits available for kinetics experiments. The first is the anabolic kinetics kit. So this allows you to label newly transcribed or nascent RNA with 4-thiouridine. It provides the iodoacetamide for the alkylation step and allows you to perform kinetics experiments to help you look at RNA synthesis kinetics. And then if you're interested in mRNA degradation or mRNA half-life, then we would recommend the catabolic kinetics kit. This one allows you to perform an extended labeling using 4-thiouridine for your RNA, and then to replace this with unlabeled uridine and then sample over a time course, which will allow you to assess the transcript stability and analyze RNA degradation kinetics. So this anabolic and catabolic kinetics kits both provide the iodoacetamide for the alkylation. In addition, they provide reagents that help to stabilize the RNA extraction process. So with the culmination of these SLAM-seq kits, you can prepare your total RNA through the alkylation reaction ready for library preparation.
Stephanie, this next one is for you too. What is the advantage of SLAM-seq over other methods utilizing 4-thiouridine?
So the primary advantage with SLAM-seq over these other methods is that you don't need to perform any biochemical pull-down, which Stefan alluded to at the beginning of his presentation. So instead of needing high inputs of RNA in the high microgram range, you only need low amounts of input RNA in the low microgram or nanogram range, and these can be obtained easily from cultured cells following the SLAM-seq labeling. And then the simple alkylation reaction means that you can also generate your sequencing libraries from a single RNA sample where you can look at the labeled nascent RNA fraction and the existing RNA fraction in one library without having to perform separate library preps.
Okay, and this next one is for you too. Can I use only QuantSeq library prep with SLAM-seq or other library prep methods? Can they also be used? What is the advantage of using QuantSeq?
So this is a really great question, and I think there were a couple of other questions that came up in regards to this. So QuantSeq is a really ideal library prep method for SLAM-seq experiments. One of the main advantages here is that you're profiling 3' tags from full-length mRNAs so that you know these mRNAs are coding for fully functional proteins. And the other advantage for a QuantSeq is that you only need 1/10 of the read depth that you would need for a usual mRNA sequencing experiment. So this means that you can multiplex more samples together, although you might need a high amount of read counts in the 20 to 30 million read range if you're looking at these short time course time points. This is still going to provide you with a significant advantage over a traditional mRNA-seq where you may need up to 100 million reads. But in principle, the SLAM-seq method itself is for preparation of total RNA ready for RNA-seq library prep. So in case you were looking at non-polyadenylated transcripts or wanted to have resolution of isoforms and sequence other parts of the mRNA transcripts, then it is possible to use the SLAM-seq generated total RNA for other library prep methods.
Stephanie, why when you post 24 hours do you change the media every 3 hours?
So the idea with changing the media every 3 hours is to ensure that the phosphouridine is remaining fresh and non-oxidized form. So the stability can decrease over time, and that's why we recommend to replace the media every 3 hours. And this will ensure that the supply of phosphouridine is fresh to the cells, and that this can be incorporated in its fully functional state so that it can be efficiently alkylated.
Next one's for you too. Hi, are there any commercially available spike-ins that are compatible with 3' end QuantSeq?
So the answer is yes. At Lexagen, we've actually designed our own set of RNA spike invariant controls called SERVs. So these are a set of 69 artificial transcripts that actually represent isoform complexity. So while QuantSeq itself is not a library prep method that is designed to capture the comprehensive transcript isoform identity, these isoforms all have a poly A tail. So these allow you to spike these into your QuantSeq experiment, and those will be generated, libraries will be generated from these SERV transcripts alongside your endogenous transcripts, and we provide a SERV suite which allows you to analyze the read counts that are assigned to these SERV spike-in transcripts, and this will allow you to generate a measure, a fingerprint essentially of the spike-in controls in your sample. So you can choose the spike-in level. We recommend spiking these in at 1%, and this allows you to assess the technical constraints of your library preparation. So we actually provide these SERV controls in 3 different mixes. One mix is our mix 1, and these actually provide 3 tubes of SERVs, and in the 3 tubes we have different equimolar mixes of some of these SERV isoforms, which means you can assess not only the isoform complexity but also the dynamic range. We have a second mix which contains just an equimolar mix of these 69 SERVs, and our third SERV set 3 contains 69 artificial SERV isoforms plus the 92 ERC spike-in controls, which also contain a poly A tail. And we actually recommend the SERV set 3, which contains both the SERV isoforms and the ERC spike-in controls, because this gives you a maximum number of spike-in transcripts available for benchmarking and control of your RNA-seq experiments.
Thank you. Stefan, this next one is for you. Could the conversion chemistry S4U alkylation be used with an RNA library prep method that does allow discrimination between mRNA isoforms and non-poly A RNA?
So in principle, yes, this is possible. So in principle, SLAM-seq is compatible with any RNA library prep method that involves conversion into cDNA. So yes, it should in principle be possible.
Okay, and this next one is for you too. Can SLAM-seq be used to quantify non-coding RNAs?
So this goes in a similar direction. Again, if one has a proper sequencing strategy, so library preparation protocol, then yes, and this involves a reverse transcription step, then yes, it should be compatible with quantifying non-coding RNAs.
Okay, and I believe more questions are coming in, but I think in the meantime it might be best if we share some closing comments, and we can always take them a little bit later. I would like to once again thank Dr. Ameres for his presentation. Do you have any final comments?
No, thank you very much for listening. It was a pleasure.
Okay, well, I would also like to thank our sponsor, Lexagen, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May 2018. You'll receive an email from LabRoots alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. We will see you next time. Goodbye.